great colleagues for more than 5 years now, I think. And he's here with the Cup Strange Fellowship, Visiting Fellowship. He's one of the leading ecologists working with food webs and you didn't say that. And metabolic experience. Yeah, I would say so. And we're very happy to have you here again. Thank you so much. And excited to hear what you have to say to us. Thank you.
Thank you, Victor. Thank you, everybody, for attending. Very happy to be back here again. It's my third time visiting UFSCar, favourite campus I've ever visited. It's great to be back and tell you a little bit more about my research. So, in terms of an outline for the talk, split up into 3 bits. The first element will be about ecosystem size structure. Here I'll be defining this term so that you get a better understanding. And this, I'll give some examples of my old research, I would call it. The second element will then start to think about environmental impacts on food webs, and that's more recent research. And then maybe if there's time, but perhaps not if people have a slow start, then I might talk about current research. Work that out. And at any point, if you have questions, I'm more than happy to take these rather than saving at the end. This is really fun. So if you don't understand anything, please say. Okay, so when we look at nature, we're faced with great complexity. The natural world, whether that's many species of plants competing for limiting resources in the forest, or complex interactions operating in the open ocean. And whether there's some general rules that underpin this complexity is what I'm really interested in. And this is because it might allow us to understand the response of natural systems to environmental change, whether that's climate change, pollution, land use change. So if we think about different types of ecosystems, then there's various things that might complicate the search for general patterns. So if we think at the base of the food webs, in aquatic systems, the primary producers are phytoplankton, unicellular algae, typically, and on land we have multicellular vascular plants. So we have a fundamental difference at the base of the food webs between aquatic and terrestrial realms. In aquatic ecosystems, the phytoplankton are fed upon a bit more efficiently by smaller consumers, such as zooplankton, which are in turn fed upon by small fish, which are then fed upon by large fish. So in aquatic ecosystems, we have strongly sized structured interactions. Big things eat smaller things that eat smaller things, with, of course, some famous exceptions. However, if we think about the same processes on land, it's slightly different. We have large multicellular plants, which means that the consumers, the herbivores, typically, of these plants can be of varying size. So they can be very large or indeed very small. And so the kind of current perception is that terrestrial systems lack this size structure. So it's, in comparison to aquatic systems, it's not necessarily large things and small things. There's much more variation on that. But if we extend terrestrial systems to think about the soils, again here, in the soils, quite often the largest organisms that you find are those low in the food web. So if you take earthworms, for example, these are quite often the largest soil invertebrates that you find, but they are detritivores, right at the bottom of the food web. So again, there's some evidence to suggest that terrestrial systems are less size structured. And if we try and think about ecosystems in the broadest sense, then of course they are linked together. And a lot of energy and matter moves from the terrestrial realm into aquatic systems, and of course vice versa. But I've highlighted this link because some of my previous research has looked at how terrestrial inputs shape freshwater ecosystems, but that's not the focus for today. So then, there's various reasons to expect that we might not be able to find these general patterns. But one approach that can be used for trying to identify similarities or differences between whole ecosystems dates back nearly a century ago now to the work of Charles Elton, who introduced this concept of ecological pyramids, that I guess you're familiar with. Here we have a pyramid of numbers, so the number of individuals in different bands that relate to size classes. And when you do this for different types of ecosystems, whether that's terrestrial, marine, freshwater, then you would typically find this kind of pattern, a pyramid pattern where there's many more small organisms than large organisms. So that was a century ago. But more recently, a common approach, at least amongst aquatic ecologists, is to use what's known as size spectrum approaches, which is essentially a very similar approach. We just take the logarithm of the body size and the abundance, and this creates a negative relationship when plotted on log scales. So for instance, we could divide up a stream food web into different size classes. That might include bacteria, algae at the smallest scale, invertebrates in the middle, and fish at the end. And for this approach, we would sum up, for instance, all the different fish individuals that you find within that size class and represent the total abundance. So in this approach, taxonomy is not particularly important. We're interested in merely the numbers of different sized individuals in the ecosystem. So what is well established is that ecological communities are characterized by many small and few large individuals. And as I said, this approach provides a lens for which we could potentially explore similarities and differences between ecosystems. Specifically, the slope of this relationship tells us something about the relative change in the numbers of large and small individuals within an ecosystem, and therefore how biomass is fluxed through the food web. So for the rest of the talk, I will be concentrating on the slope of these relationships to explore the differences that we might be able to find between ecosystems and the effects of environmental change. Any questions at this point? No? Great. So, to prove that this approach applies in more recent research, I'll show you a couple of examples from some work that I've been involved in. When we look at the base of the food web, as I said, primary producers differ in size between land and in water. Here we have the size spectrum, or many size spectrum, in fact, for 242 locations across the globe, where researchers, not me, but other researchers, have been out measuring the size of trees in forests, measuring the diameter at breast height as a standardized measure of the size of individuals across transects or within plots. So we can see that trees follow this pattern. There are many small trees in the forest and few large trees following this parallel. We've also done the same analysis for phytoplankton in the ocean, unicellular algae. Again, I haven't been involved in collecting these data, but there are lots of data sets where researchers have looked at the different size of the algal cells and their abundance. And when we do this, we see, again, a very similar negative linear relationship that is, at first glance, strikingly similar to what we find on land. But I put similar in brackets because there's subtle nuanced differences if we compare the exponents of these relationships. So the exponents are slightly different on land than in water, and I'm happy to talk about why this might be at a later point, but in the interest of time, I'll move on. Even though to say that when we do some corrections, then actually these relationships are very, very similar.
And closer to the 3/4?
Yes. Yeah. So from metabolic scaling expectations, then the prediction would be -3/4, which we see amongst phytoplankton And we do see in trees, but when we look at diameter, not mass. Okay, so that's within a single trophic level, but what happens when we look at across trophic levels? So individuals that interact with each other through trophic interactions. So I'm an aquatic biologist by training, and this is a data set that I've been working with for many years, rinsing it as much as I can, where we've looked at the size structure, or size spectra of freshwater streams in the UK. Again, we find this linear negative relationship, and this is going from small invertebrates to large predatory fish. In this paper, we were interested in looking for consistent patterns across different types of ecosystems. So we managed to compile similar data for different ecosystems, marine rock pools, which include a global data set spanning from the equator to the poles, and for terrestrial soils. So in the marine rock pools, we have small invertebrates up to small fish, and in the soils, it's invertebrate-based food web. So again, from a first glance, we're faced with this common pattern, linear negative relationship that applies within ecosystem types. And actually, if we look at the edge of this panel plot between the freshwater streams and the soils, there's what I would say remarkable similarity in the size structure there, indistinguishable exponents from the physics models. But when we look at the marine rock pools, we see a slight deviation from this in terms of they tend to have shallower slopes, which indicates that there's relatively more larger individuals than what we find in the other systems. And I guess this is perhaps unsurprising when we think about the type of ecosystem that rock pools are. They're not a closed ecosystem. Two times in a day, tide comes in and out, replenishes the ecosystem with perhaps top predators that actually feed upon a much larger spatial scale than what is sampled within a given rock pool. So perhaps if we applied a similar approach but looking at a larger spatial scale, then we might find greater convergence between ecosystem types and between expectations. Okay, so that's a very quick tour of my previous research that's kind of looked at these general patterns across ecosystems. But as I said, what I'm really interested in is understanding how this approach could potentially be used for measuring or quantifying the effects of environmental change in different ecosystems, and therefore allow an approach that might be used for monitoring. So in 2010, a very small paper written by Owen Petrie and Andrea Belgrano summarized the state of play in terms of size spectra research and highlighted that perhaps body size distributions and size spectra in particular can be used as a measure of how stressed an ecosystem is given different environmental pressures. This is because a review of the literature at that point seemed to indicate that there was a systematic change in size spectra between reference conditions, so pristine ecosystems, and impacted ecosystems which are under stress. Specifically, relatively fewer large individuals are found in impacted ecosystems or sites. And this is, I guess, intuitive when we think about the differences that occur, that are correlated and occur with body size. So larger individuals have a lower population density to start with. As I've shown, we have this negative linear relationship. So small populations are more prone to stochastic processes, including extinction. Large organisms have a large home range size for which they acquire their energy, and we can imagine that certain types of environmental impacts, fragmentation, land use change, for example, can be particularly strong for larger organisms for which operate at large spatial scales. And large organisms have slow generation times in that they inherently probably have a lower capacity to adapt to environmental change because of the intrinsic reproductive rate that they exhibit. So for these reasons, perhaps size spectra can be used as a measure of how far ecosystems are away from the baseline that I've kind of shown you already. Specifically, what my recent research has been interested in is understanding the impacts of land use change and how this, and whether this concept of size spectra, whether they show systematic response to land use change occurring at different locations. So here we have simulated images of land use scenarios in upland Wales in the UK, where systems have been transformed from more natural states to quite high intensity grazing with a reduction of forest, or indeed where forest occurs, it might be plantations, exotic plantations. So what impact does this have on stream size spectra? Of course, streams are the veins that flow through the terrestrial landscape and are largely affected by in Basque Country in Spain, where we've been interested in looking at how the stream size spectra change across different catchments. So catchments where you have native deciduous forest and catchments where you have plantations. Forestry plantations, specifically eucalyptus, which is a common species grown there for timber, etc. And as you can see from the plot here, we have our native forest represented in green as our, let's say, reference or a natural state, and plantations highlighted in red as the impacted altered state. We can see that we have a shift in both the elevation, that's the most striking pattern, that's the red data points are much lower than the green data points, so there's many fewer individuals found in the plantation streams than there are in the native forest. And there's a change in the slope as well, where we have relatively fewer large organisms compared to small organisms. So this is somewhat consistent with those expectations, those characteristic effects that I mentioned. So in this system, we find a lower abundance of large invertebrates in the impacted streams. And what was nice here is that we managed to demonstrate an impact at the ecosystem level. So the size spectrum slope in this case predicted quite strongly the decomposition rate of organic matter within those stream ecosystems. Essentially organic matter, so the leaves and vegetational material that falls into the rivers is breaking down by a range of invertebrate species. And this demonstrates that the large species are the important species within that ecosystem for breaking down that detrital material and therefore for carbon cycling. This is quite a nice example between how we can link organizational levels from community to ecosystem levels using this approach. So here we find a lower abundance of large invertebrates and reduced functioning in the impacted streams. Again, another study that I was lucky to be involved with, led by Victor and master student Giovanna Collier, here in Oscar, looked at a range of different streams in Brazil, looking at the invertebrate community across native to agricultural ranges. And what's really interesting for me, working on these two papers in parallel, that both came out in the same year, but finding quite different patterns in two different locations. So, whilst we find a similar pattern in terms of the elevation, it's much lower in the agricultural sites. So again, these are on a logarithmic scale. So here we have a massive difference in the abundance of organisms that you find in the agricultural sites. We actually find slightly shallower slopes. So this indicates relatively more larger organisms relative to small than what we would expect based upon the body size. And in this paper, specifically, Perkins put forward the idea that perhaps these more impacted sites have higher energy transfer through the very simplified food webs that occur. So along this body size gradient change, we can also find a great change in biodiversity moving from native to agricultural streams. And perhaps these shallower slopes is indicative of relatively higher energy transfer from small organisms to large organisms in these very simplified, relatively simplified, at least compared to temperate systems, food webs. And so this is something that I'll come back to at the end. Okay, so size spectrum approaches can be useful because they apply across different types of ecosystems. They can potentially be used for measuring environmental change. But what about reversing the impacts of environmental change? Can they be used to monitor ecosystems returning back to a less impacted state, or let's say back to a pristine state? And some of my recent research has looked at this notion of ecological restoration in attempting to restore ecosystems, replenish them back to a previous state before environmental change. A nice quote by E.O. Wilson, at the turn of the century, said, "Restoration is the means to end the great extinction spasm. The next century will, I believe, be the era of restoration in ecology." But like most of his research, he wasn't wrong, at least in aquatic systems. River restoration is now a multi-billion-dollar industry, at least in Europe. I don't know, this is perhaps a nice discussion point and idea to think about between temperate and tropical locations, but in temperate Europe, a lot of money is currently being spent on trying to restore ecosystems, river ecosystems back to their former glory. So that might be introducing large woody debris to diversify the flow, fish passes to allow fish to pass what used to be weirs, simple, perhaps the most simple restoration technique, fences. This is an aerial shot of River Skjern in Denmark. You can see the impact of putting fences that reduces the grazing close to the river, but also perhaps heavy modification, meandering the river back to the former shape so it has better connectivity between the land and the water so that these processes can be rejuvenated. And I've been involved with working on a restoration project in my local river to my university in London. Specifically, this is Beverley Brook, which is situated in South London, that flows into the River Thames, the main river that flows through London. And so this system is a typical urban stream, has poor water quality, and has suffered from generations of simplification. So straightening, narrowing, removing vegetation to try and move it from here to here as quickly as possible. The name Beverley Brook actually translates in Old English to Beaver's Meadow to suggest that this guy up here used to be very common, at least 500 years ago or so in this catchment. And beavers are ecosystem engineers in terms of leading to diversification of habitats within aquatic systems, so that you have deeper pools and riffles and heterogeneous environments. So in the absence of natural engineers like this, this means that we as humans have to go in and modify the ecosystem. I want to show you a short video now of the before and after of the stream 3 years apart. So on the left-hand side is the stream before any restoration work. You can see that it's very straight very shallow and very silty. There's very little flow and very little connectivity between the land and the water. On the right-hand side, hopefully you can see it with a very different picture. In terms of, we have much faster flow. The simplest approach is to narrow the stream, gives it more energy, that moves the sediment quicker. Landscaping it so that you have greater connectivity between the marginal plants and the river. And introduction of large woody materials such as this and this creates pools and scours, diversity of habitats.
It's curious because in my former department,
Yeah.
you mentioned that in Brazil or global south, the restoration efforts are things like gardening. They're not restoring the existing function of the streams, just the existing building.
Yeah.
Sometimes we improve the diversity, but the stream is not functioning well.
Yeah.
And this is really true that we see here in Brazil.
Yeah, I mean, it's a similar story to most of the work in Europe, to be honest, in that it's perceived as gardening. In that you might make it look nice, but there's very little time and money spent on monitoring whether it has any effect. Right? So quite often things are done because they look good, but you have no idea whether they're effective. Yes, reinstating the ecological processes requires perhaps larger scale intervention. For example, landscaping and connectivity between the water and the land is incredibly important. But I would say there's still limited evidence to demonstrate improved functioning. Yeah. So, yeah, okay, well, at least I'm noticing, so does it work? So, as part of this project, with my students year on year, so I get involved in this project in 2015 and still work on it. Every year I take the students out on field course and I have undergraduate students doing their project. And doing this with monitoring. So we have found that there is greater species richness in the restored sections and after the restoration, but this is restricted to the fish assemblage, not the invertebrates. Generally, there's larger body size within species, but again, this is predominantly within the fish assemblage. So 3 common fish species in particular are much larger in the restored section than in the control section. And there's about 282% higher fish biomass in the restored section than in the control section, which is quite a massive amount. The invertebrate change was also large, it was over 100%, 110% larger in the restored section. So in terms of fitting it back into the scheme, then essentially what we've seen is a transition from this situation to this situation. Relatively more larger individuals back in the ecosystem, an increase in the elevation, so numbers of individuals is greater, but limited response to biodiversity. Along with biodiversity is the ecosystem processes, so that is something that we haven't really looked at. I have another couple of videos that hopefully show this. Do you have this phrase in Portuguese?
What?
A picture is worth 1,000 words. Yeah? Hopefully. So this is me with my GoPro after doing some electrofishing. And this is releasing the fish back into the stream after we've done the fish surveys. So this is the control stretch. You can see that it's very sandy, silty, murky. Lots of very small individuals. We have things like stickleback, minnows. That's a European eel. That almost had second thoughts about going back up there, but we'll go the other way. Then you contrast this with the restored section. So large cyprinids, this is barbel. We found, but this is a species that we didn't find in the control reach. So they're very large at the top of the food web. Another common cyprinid chub are much larger in the restored section.
Do you have a pike?
No, but we did find perch, which is a skipper's fish, yeah. So then we have much, as you can see, generally larger and more diverse fish assemblage going back in. Again, some eels, which are actually globally threatened at the moment, so that's good to see. And a flatfish. That's a flounder, which is actually a marine species, but the river is very close to the River Thames, as I said, which is tidal, so they do make their way up into the freshwater.
You know, aujourd'hui, we're river.
Which rivers are
Aujourd'hui, river. Grégoire River. Sometimes. Yes. C'est intéressant.
the one that goes through the city?
Yeah, maybe the one that's just behind your hotel.
Yeah, that smells.
Yeah, it smells bad.
At the moment, it smells bad, yeah. I remember last time I came here, it wasn't like this. But
Yeah, we should get some big brands and restore this.
yes. Yeah. After you. Okay, so how are we on time? Okay, then I will tell you a little bit about the current project, but very briefly. We've already covered a lot, so it'd be nice to talk a little bit more. So, thinking back to land use change, this is a pressure that's felt across the globe. But I would say there's a long history of change in let's call it the global north. So temperate locations such as the UK, where I live, have gone through land use conversions a long time ago. This is a very famous picture by Constable showing an agricultural scene in the 1800s. Contrast that to perhaps developing tropics, where conversion of rainforest to oil palm plantations or sugar cane, for instance, is occurring at a very rapid scale. So I guess the challenge here is to develop a general framework to be able to predict what these responses should be across latitudes. And hopefully I've got the idea that body size distributions are probably going to be a good approach for this. But we still need to do a little bit more work. And through collaboration with Victor, wow, that's 3 years ago already, we had a paper in Trends in Ecology and Evolution, where we were trying to think about how the assembly of ecological communities might differ in temperate and tropical locations, let's say, but generally speaking across latitudes. And of course, one of the most striking differences with latitude is temperature. And in this paper, we were drawing upon principles of metabolic scaling theory, but also ideas about assembly processes, so stochastic and niche processes. So if we just think about temperature, of course, much higher in tropics or subtropics than in temperate systems, temperature drives lots of biological processes. So the mortality rate increases with temperature and metabolic rate, lifespan decreases, population size should decrease, given a fixed amount of resources, and at higher temperature, everything happens quicker, so biomass turnover should be greater at higher temperature and higher metabolic rate. So, from these simple ideas, which in themselves follow consistent scaling and power relationships, we might expect that there should be marked differences in the size structure, size spectrum of communities, of temperate communities, which are represented at the top there, and tropical communities in red. So specifically, if we think about these four processes, then ultimately this suggests that the turnover of organisms is higher at higher temperatures, and therefore the standing biomass for that system will be lower. Everything's operating at a faster speed. So we might expect a systematic difference in the elevation, so the intercepts of the relationship. Also related to metabolic principles, temperature size rules would suggest that we're tropical locations going to have smaller individuals on average than in temperate locations. And we, but specifically Victor, made a suggestion that in the tropical location, because of the small population size and rapid biomass turnover, they're more likely to have stochastic processes than in temperate communities, which are more driven by niche processes. So perhaps these stochastic processes operate also amongst the trophic interaction level, leading to more variation in the consumption of prey by predators, and therefore more variable size spectra from time point to time point, or even for a given time. Along with this, and we suggested this in the paper, but subsequently this has kind of been shown experimentally, that the transfer efficiency of energy decreases with temperature as well, which might mean that we're going to see a difference in the slopes. So at higher temperature, because of the higher cost of growth, then the amount of energy that can be transferred from smaller organisms to larger organisms is relatively lower, so we have steeper slopes in tropical locations. So perhaps these subtle differences between temperate and tropical locations might explain the differences that we observed, or the unexpected patterns that we observed between the temperate example that I showed you earlier and the tropical example related to land use change. So this leads to the question, is the impact of human pressure on food webs different across latitudes? And to address this, we, myself and Victor and a couple of Tina, have led a working group, data synthesis working group, that has tried to address this. That's funded by Aquasync, which is a data synthesis centre in Denmark, which have kindly funded 2 nice workshops, represented by the different pictures there, that includes Francisco and Victor and many others, where we've spent time trying to collate data sets from across the globe to address this question. So this is perhaps not the most up-to-date version of the map, but we have quite a good global distribution of data for invertebrates and fish body size across latitudes, but specifically in the Americas, I would say. So we have 44 data sets and about 9,000 sites. So we should have some nice power to be able to look at some of these ideas. So to standardize things, then we take an approach where we're using human footprint index, as it's known, as a metric to quantify the human pressure across the globe. So we're using global maps such as this that rank different human pressures, 8 different pressures ranging from buildings to population density, nighttime lights, vicinity to roads, railways, waterways, etc., to quantify human pressure for each of the sites within the data set. And this is an index that ranges between 0, where essentially we have wilderness, where there's no human pressure, to 50, which are very modified and corporate systems. So results, ongoing. That's one of the things that we're spending this time visiting Victor to think about and work on a little bit. And I hope at some point I can come back and tell you about the results of this. But yeah, hopefully that will be the case one time. But that leaves me to say thank you and to acknowledge everybody who has funded this collaboration with Victor. Thank you, Victor, for hosting me yet again. Looking forward to going out to the field this week. And yeah, thank you for the funding bodies and thank you for your attention. Happy to take questions.
Questions? In Portuguese?
Yeah,
I have the fine. I have one from the beginning.
Yeah.
So you showed the whales there and how they, it's like an outlier in terms